Our next speaker is Yoram Cohen. Dr. Cohen is the head of the Fertility Preservation Research Lab in the Department of OBGYN at Tel Hashmor, Israel. He's a senior lecturer at Tel Aviv University. He's been interested in understanding the discovery of genes involved in ovarian cancer, and he'll talk to us today about, are we close to solve the mystery of Fragile X-associated premature ovarian insufficiency in FMR1 Premutation carriers?
I would like to thank the organizing committee for letting me speaking about this important subject, and I will try to elucidate some new information regarding the etiology of Fragile X premature ovarian insufficiency. A remarkable progress has been made to identify a constellation of clinical significant disorder associated with dynamic range triplet sequence mutation affecting the X-linked gene, also known as FMR1. The fully extended form of the mutation leads to a Fragile X syndrome, the most common cause of inherited mental retardation, as well as the most common known genetic cause of autism. Affected individuals with mental retardation had more than 200 hypermethylated CGG repeats in the 5' untranslated region of the gene. The hypermethylated expanded CGG repeat shut down the promoter, and therefore no messenger RNA is produced, and the corresponding product of the gene, the FMRP, is absent, which leads to the Fragile X syndrome. Allele within the 6 to 55 triplets are considered to be normal, and those with 29 to 30 triplets being the most common in the general population. Allele with 55 to 200 CGG are considered to be premutation. They are transcribing the protein is produced, but these alleles are unstable and can progress to full mutation, expand to full mutation upon maternal transmission. The term premutation was applied to the range of there is something that is called the gray zone, which is the 45 to 54, which is the intermediate. This repeat size is usually it's stable, but it could be expanded, not to the full mutation, but could be expanded. AGG interrupts within the CGG tract are commonly seen in the FMR allele, and normal allele frequently have 2 AGG interrupts. AGG interrupts spaced approximately 10 repeats apart are associated with reduced risk of expansion and do not influence the FMR1 messenger RNA level. This important AGG interrupt is important in genetic consultation when estimating the risk of expansion. The risk for expansion is dependent on the CGG, is governed by the CGG size of the maternal allele and the number of AGG interrupts. Maternal allele in the range of 70 to 80 CGG with 2 AGG interrupts are about 8 times less frequent to progress to full mutation. And this interrupts reducing the risk in comparison to the same CGG range without the interrupts, which is about 8 times higher. And here we can see that in about 80 CGG, this effect is lost, mainly because the AGG is not present in a more expanded tract. Sorry. Okay. Repeats with 55 to 200 are termed permutations and are common in the general population, with estimate prevalence dependent on the population assessed. In Israel, roughly 1 in 150 female carrier, in 150 females is a carrier. Historically, women who harbor a permutation allele were thought to be clinically unaffected by the expanded allele. However, it is clear now that multiple distinct clinical phenotypes are common among carrier females and among carriers, and at least 2, the Fragile X primary ovarian insufficiency and the Fragile X tremor ataxia syndrome, are unique to this permutation range. Sorry. So I've lost myself here. Okay. Fragile X-associated tremor ataxia is first described in 2001 by Randy and Paul Hagerman, who noticed that grandfathers of affected children have a neurological problem, and this neurological disorder, which is the core feature of it, is intentional tremor, gait ataxia, and some intellectual disabilities is more common in this. Signs of this neurodegenerative disorder are brain atrophy, specific MRI sign that usually showed in the middle cerebral peduncle, and neuropathy. And by pathological histological, a specific intranuclear granules are shown in the nucleus. The symptoms of FXPOI usually develop in older ages, and the average age of onset is 60 years. FXPOI is more frequent in males and affecting about 45% of the males and 8% of the female carriers older than 50 years. The symptoms appear to be more severe with increased CGG, with a strong correlation to the age of onset and the age of death. Prior to the discovery of the FMR gene in 2001, Chronister et al. Reported a 20-fold increase in the incidence of premature ovarian failure, defined as cessation of ovarian function prior to the age of 40. Interestingly, women who are with a full mutation have the same risk as non-carrier, which is 1%, suggesting that different underlying pathological molecular pathologies are responsible. the term Fragile X associated ovarian insufficiency should be better describe the spectrum of clinical and laboratory features of the affected FMR1 permutation carriers. FXPOI include absence or irregular periods, symptoms of reduced fertility, such as longer time to first pregnancy, seeking more fertility help, and increased level of day 3 FSH and decreased levels of AMH. Most of these patients also experience menopause earlier by 5 to 7 years. The repeat size are strongly associated with ovarian insufficiency, but in non-linear way. The risk of ovarian dysfunction, defined by both the prevalence of premature ovarian failure or age of menopause, is showing in all the patients, but in particular in patients in the range of CGG between 70 to 120 repeats. This curvilinear association between CGG repeat length and fertility has been demonstrated in other reproductive aging milestones, including menstrual cycle alterations and in vitro fertilization outcomes. So what could explain the different phenotypes? In contrast to individuals with full mutation, in whom the hypermethylation of the expanded CGG leads to transcriptional silencing and absence of the FMRP, in carriers, premutation repeat expansion, the FMR gene is unmethylated and remains fully active, and actually the gene expresses increased levels of FMR1 messenger RNA, which is thought to be the result of this toxicity. Several studies have shown that premutation alleles are characterized by high level of messenger RNA expression that could be increased by 2 to 8 folds relative to the normal controls. In a recent series, 800 males were assessed, and all of them has the full range of the CGG repeat, and the results show that FMR1 messenger RNA levels increased with the increase of the CGG repeat number, and that significant increase was detected from allele length as short as 35 repeats. In an elegant series of nuclear run-on experiments, Flora Tassone and her colleagues showed that the elevated level of messenger RNA in premutation carrier is caused by increased transcriptional efficiency rather than increased RNA stability. Furthermore, Tassone has demonstrated that the AGG interrupt, which protect from expansion, does not influence the FMR1 messenger RNA levels. Despite the higher level of FMR1 transcript, mild deficit of FMRP have been demonstrated in premutation carriers. Therefore, FMRP deficiency is probably not the principal cause of FXPOI and FXTAS. The crucial observation that RNA containing expanded CGG repeats accumulate in nuclear RNA aggregates in the brain section of patients with FXTAS support the notion that elevated levels of FMR1 RNA trigger the neuronal toxicity. But what is triggering the toxicity in the ovary? Do the FXPOI and FXTAS share the same molecular basis? A recent study by Liu and his colleagues characterized a mouse model carrying a FMR1 permutation allele and show that FMR1 permutation RNA is sufficient to cause reduced number of growing follicles and affecting the number of pups. FMR permutation mouse deliver their first litter later as compared to the wild type, and the average number of pups was significantly reduced. The permutation mouse models support previous data that FMR1 permutation does not impact the establishment of primordial follicular pores, but rather implies the development of the growing, it is affecting the development growing follicles. We can see that in younger pups, okay, the weight of the ovary and the number of oocytes is similar. However, in older pups, we can see that there is a difference. Also, the hallmark of the FXPOI in humans is elevated FSH, and this is also was demonstrated. And something that was very interesting is that the LH receptor was strongly downregulated. The AKT and the mTOR pathway are very important in ovarian physiology, and actually knocking down several of members of this pathway can result in premature ovarian failure. So in this model, they show that a significant reduction in the phosphorylated AKT and also the phosphorylated mTOR, but not the proteins, just the phosphorylated form of the protein. To examine for the first time the effect of elevated FMR1 permutation mRNA in human, we assessed the role of RNA accumulation in granulosa cells obtained from permutation patients undergoing IVF PGD treatment, and compared it to the granulosa cells of women undergoing IVF for male factor. 21 permutation carriers and 15 controls were evaluated. There were no difference between the two groups regarding the patient age, parity, duration of simulation, number of embryo transfers, or pregnancy rate. But in accord with previous study, the permutation carriers observed higher FSH, higher FSH and FSH to LH ratio, and also reduced response to controlled ovarian hyperstimulation. The FMR permutation carrier, when we examined it, we found a significant nonlinear association between the number of CGG and the number of retrieved oocyte. And the nadir of this, the lowest number of oocyte was retrieved in women in whom the CGG repeat was in between 80 to 120 CGG repeats. A trend of nonlinear association between the number of CGG repeats and the level of messenger RNA in the granulosa cell were also noticed. And again, this association was higher in the group of women that harbor 80 to 120 CGG repeats. We observed a significant negative linear correlation between FMR1 permutation level in granulosa cells and the number of oocytes retrieved. Collectively, our data support the notion that elevated levels of FMR RNA might elicit ovarian dysfunction and FXPOI. We also compared the permutation patients between 80 to 100 and 20 to the permutation with lower and higher CGG, and we noticed a significant difference in the oocyte retrieved and also in the messenger RNA. These findings are in accord with the curvilinear association between the CGG repeat length and fertility in FMR permutation carrier demonstrated in previous studies. The AKT and mTOR pathway, as mentioned before, are required for proper control of ovarian function, and inhibition of mTOR can result in reduction in granulosa cell proliferation and survival. Given the observation of FMRNA accumulation in granulosa, we assessed this pathway, and we found a reduction in the AKT in compared phosphorylation in compared to the control, and also the same in the phosphorylated. It was reduction in the mTOR between the FX premutation to the control. To date, many studies have suggested that repeat-associated diseases can elicit their toxicity via RNA, presumably by binding and sequestration of specific RNA binding proteins. This RNA gain of function mechanism has been best established in myotonic dystrophy, in whom the CUG repeat expansion in the 3' untranslated region of the DMPK bind and sequester the MBNL family of RNA binding factor. Depletion of the free pole of the MBNL1 lead to dysregulated splicing and therefore triggering the symptom of myotonic dystrophy. Extending this RNA gain of function model to FXTAS and FXPOI, the extended CGG repeat are predicted to sequester specific proteins, resulting in loss of their normal function, which ultimately can cause symptoms of FXPOI and FXTAS. In FXTAS, CGG repeats has been shown to avidly bind a large number of proteins, including SAM68 and the microRNA biogenesis complex DROSHA and DGCR8. In attempt to elucidate the pathogenesis of FXTAS, a recent study analyzed the protein that bind to the pathologic CGG repeats expansion. Proteins were captured using biointegrated RNA composed of non-pathogenic 20 CGG repeats or pathogenic expansions of 60 to 100. The DGCR8 and his partner DROSHA were preferentially recruited to the long CGG repeats. DGCR8 presented in the normal a diffuse pattern. However, in the abnormal expanded, the DGCR is also located together with the CGG, meaning that they are captured by the CGG repeats. When this observation has been examined in Faxtal's brain section, a sequestration of the DGCR8 and its partner drosha within the CGG RNA aggregates was shown, unlike the normal brain tissue. In here, we can see the intranuclear aggregate, which is stained for the CGG and the DGCR, meaning of the sequestration, abnormal sequestration of the DGCR8. The researchers proposed a model that this sequestration affecting the microRNA processing in the cell and thereby extracting their pathological effect. Ablation of the screening studies for RNA binding proteins also found SAM68 constantly colocalized with CGG RNA aggregates. As seen in the slide, the same pattern that has been shown for the DGCR8 is also shown to the SAM68. However, Sellier and her colleagues were able to show that it is indirect connection, but it is through protein-protein interaction between the DGCR and the same 68, which is what is happening. In accord with the RNA gain function mechanism, where the CGG repeat expansion RNA bind and sequester SAM68 protein, depletion of the free pool of the protein leads to functional ablation. We were fortunate to find a paper that was published in 2010 by Bianchi and her colleagues, reported the functional consequences of SAM68 knockout mice and female infertility. SAM68 directly bind, they found that SAM68 directly bind to FSH and LH receptor in a method that was controlled by FSH. When we looked at the fertility outcome, we have noticed that the cumulative PAPs that was reported previously in the premutation mouse and in the SAM68 is the same. Encouraged by this similarity, we also compared the phenotype. Amazingly, premutation mouse model and SAM68 knockout mouse had very similar fertility phenotype. Both mice exhibit impaired female fertility, sterility rate, reduced number of PAPs, reduced number of growing follicles at the age of 9 weeks, and this is suggesting that the SAM68 probably is related to the FXPOI phenotype. Todd et al. Have recently proposed an alternative model of FMR premutation toxicity. In this model, toxic peptides are produced through a process called repeat-associated non-AUG initiation translation. In this model, translation of expanded CGG, translated of the expanded CGG does not occur through the normal AUG. However, this is giving a rise to a cryptic protein, and the most common one is the poly G1. Staining of brain tissue and cardiomyocyte and various other tissue showed that this poly G accumulates in these nuclear aggregates. Recently, it has been shown also in the ovary. So we think that the RAN translation might also relate it to this phenotype. I'm coming to the end. Long non-coding RNA are one possible mechanism that may involve in regulation of gene transcription and translation. And recently, Pastore has reported the discovery of long non-coding RNA named FMR6 that originate in the antisense of the FMR gene. FMR6, he found that FMR6 was downregulated, was reduced in premutation carriers, and suggested that abnormal transcription toward the distal end of the FMR1 locus is also involved. Prompted by this observation, we analyzed 22 permutation carriers and 11 controls, and found that there was no differences in regard of patient age, parity, duration of stimulation. But in accord with previous study, we show that high levels of basal FSH and reduced response to control hyperstimulation. Interestingly, we have previously reported that the FMR1 are we have previously reported for the FMR1 there is a significant nonlinear association between the number of CGG and the granulosa and the repeat. And the granulosa cell FMR6 expression level, women in the mid-range of 80 to 180 had significantly higher granulosa cell FMR6 messenger RNA, and the FMR6 transcript levels were significantly negatively correlated with the number of oocytes retrieved. I would like to thank all the people that was part of this extensive work, mainly our IVF unit and our collaboration from French and from other places. And we are making a conference regarding this subject in 2017 in Jerusalem, Israel. Thank you very much.